Hello guys and welcome back to Excelsior Academy. So in the previous lectures, we all talked about the membrane solute transport where we have mentioned about the passive transport, right? Now from today onwards, it will be all examples of active solute transport. So it means active means we are going to use ATP, energy is being used. Okay, so it is the movement of molecules through a biological membrane from lower concentration to higher concentration with the help of ATP hydrolysis. So please remember, from low टू हाई वी आर गोइंग, हम अगेंस्ट द कॉन्सेंट्रेशन जा रहे हैं, ठीक है. एंड ये करने के लिए हमें एटीपी हाइड्रोलिसिस लगेगा. सो द बाइंडिंग इन द एटीपी हाइड्रोलिसिस इट ब्रिंग्स कन्फर्मेशनल चेंज व्हिच रिजल्ट्स इन द ट्रांसपोर्ट ऑफ मॉलिक्यूल्स. It is a vital phenomena for maintaining the balance of ions across the membranes by concentrating metabolites in a certain organ or the compartments, and also by exporting waste from the cells. For example, exporting waste, any cell which has got the waste, or suppose let us say that the toxin has entered. Okay, now certain cells, they have got the mechanism that they can prevent its action, that is the killing of the cell by the toxin can be prevented if the organism has got the pumps which can take these toxin out. So अभी क्या हो रहा है, टॉक्सिन आपकी सेल के अंदर कम है और यहाँ ज्यादा है, करेक्ट. If any toxin is present, obviously outside it will be high and inside it will be low because of its property. Maybe it is soluble in lipid and all, it might have gone inside, but it is toxic to the cell and the cell understands. Okay, now we don't want this toxin to be present inside the cell, which is obviously in the low concentration but might result in the death of this particular cell. अब cell क्या करेगा, with the help of these pumps and ATP, it will take and pump out these toxins from inside to outside. Since it is against the concentration gradient, it needs ATP. So this is one example. So like this, many more we'll be discussing. So is this concept clear? Like how ATP based active transports are there?
Yes, ma'am.
Okay. Now, as I mentioned before also, the concentration of Na plus, K plus varies from outside and in the cytoplasm. If we see in this figure, you can see that Na plus is high outside, यहाँ पे कम है. K plus बाहर कम है and अंदर ज़्यादा है. Likewise, chloride also. Okay. So Na plus यहाँ ज़्यादा है, so we can expect chloride also to be more here. Together they maintain the osmolarity. Then calcium, if you see, even though it is less, but compared to this 10 millimolar and 1.2 millimolar, which one is highest? This is highest. Okay, so to maintain this concentration gradients in the extracellular space and in the cytoplasm of the cell, we need ATP based active transport. Okay, now, so the transmembrane transporters which is associated with this energetically favorable transportations are named as ATPases or simply pump. जब हम कोई चीज लो को high concentration gradient में डालते हैं, हम उसको pump बोलेंगे, ठीक है. Now we have got three types are there based on the structure and functions. So P type ATPases, F type ATPases, and V type ATPases. So we'll talk about these all. So let's talk about first P class pumps. So P class pumps, they are made up of two large identical catalytic alpha subunits which carry ATP binding site. Okay. One of them get phosphorylated at aspartate residues. Please guys, this is important. Okay. During the transport. They also contain two smaller beta subunits concerned with the regulatory function. So कुछ ऐसा रहेगा. आपका alpha and beta है. Okay, now aspartate residues of this alpha gets phosphorylated. Now all P-type transporters have similarities amino acid sequence, especially near the aspartate residues, which undergoes phosphorylation. Now it can be inhibited by the phosphate analog called vanadate. अगर आप structure देखो vanadate का, it is similar to your phosphate, right? If you know the structure of the phosphate, हम कैसे बनाते हैं? PO4 minus बनाते हैं, correct? If you see the structure of the phosphate, then you can see that the vanadate and the phosphate, it has got the same similar structure. We can't say same, sorry, but it has got the similar structure. Okay, it is almost similar. If P is over here, then same. Yeah, आपका hydrogen bond, then this one, and over here. Okay, so this is your phosphate. This is exactly similar to your vanadate, and that's why vanadate is able to act as the inhibitor. Okay, now bacteria uses P-type ATPases to pump out toxic heavy metals ions such as cadmium 2 plus and copper. So if these cadmium and copper, they go inside of the bacteria, it is going to cause toxicity in the bacteria, which results in the death of the bacteria. Okay. So if they are resistant, it means what? They have got these P-type ATPases pump to pump out all these heavy metal ions. So it means what? These are the bacteria, these are the bacteria of the class where the heavy metal toxicity cannot work in order to kill. Clear? Are you able to follow? Now, let's talk about the examples which comes under the P-type ATPases. For example, calcium 2 plus ATPase. This is one example, okay? It is a form of P-type ATPases which transfer calcium after a muscle has contracted. The two kinds of calcium ATPases are plasma membrane calcium 2 plus ATPase PMCA and sarcoplasmic reticulum calcium 2 plus ATPase. क्या ये सुना हुआ लग रहा है? कहाँ पे होता है sarcoplasmic reticulum? Have you heard about this type of endoplasmic reticulum? कुछ सुना हुआ लग रहा है?
Yes, ma'am.
कहाँ पे
Yeah,
सुना
in
है?
muscle.
Yes, correct. Any other features you can tell about sarcoplasmic reticulum? What is the main sorry? What is the main function of sarcoplasmic reticulum in the muscles? So usually,
Think it is
yes.
storage of calcium.
Yes, correct. It is actually acting as a storage of calcium, but since we are talking about the muscle contraction, so in that case, this sarcoplasmic reticulum is actually helping in transmitting the electrical impulse also, along with the calcium storage. Okay, so this is the main role. We'll talk about this in the animal physiology section. No worries. Okay. So once we are talking about the calcium transfer, okay, so these are the ATPases which is helping in pumping out the calcium. Okay, and but the regulation. So when we say regulation means what? Like who is responsible for checking the concentrations of the calcium inside the cytoplasm or outside the cytoplasm? It is these calcium binding proteins called calmodulin. Okay, please remember this is calcium binding calmodulin proteins. So cytosolic calcium binding protein, the calmodulin triggers the export of the calcium 2 plus ATPase to maintain the low concentration of calcium that is in the resting cell characteristics. Okay, now muscle cell has contracted. Contract हो गया तो मतलब calcium 2 plus आपका release हुआ है. Okay, understand this. Contraction means calcium 2 plus has released. Now release means the concentration has gone up. So the calmodulin proteins, there is so much concentration of calcium in the cytoplasm, correct? So these calcium can go and bind to the calmodulin. What are calmodulin? They are cytosolic calcium binding proteins. So this triggers the response to the cell to increase the formation or synthesis of the calcium 2 plus ATPases. So once these are exported to the plasma membrane of the calcium muscle, what will happen? These calcium 2 plus will be pumped out and the cell will come to its resting stage. Are you able to follow? यहाँ पे क्या हुआ scene समझ में आया? Should I explain once again? See, so itna to samajh mein aa gaya that under P-type ATPase we have got calcium 2 plus ATPase. Under calcium 2 plus ATPase we have got these two type of calcium 2 plus ATPase, right? PMC and SERCA. Now please understand like how it is working. So for muscle contraction, there is a calcium release. Okay, calcium release hua, so it means in the cytoplasm the calcium level got increased, correct? Now, since it is increased in the cytoplasm, there are so many possibility of binding of this free calcium to the calmodulin. Agree? So these free calcium can come and bind to these calmodulin. Now the binding of the calcium to the calmodulin triggers the export of the calcium 2 plus ATPases. Now this will go and get exposed on the plasma membrane like this of the muscle cell. Correct? So they are going to take the calcium जो यहाँ पे excess है cytoplasm में outside and will bring back the calcium to its resting stage. Sorry, it will bring back the cell into its resting stage. Now it is clear? Yes, any point anywhere which is not clear? Tanvi, samjha?
Ma'am, repeat that last what you explained.
Okay, so at the
Resting state.
so what is happening is that, see, the calmodulin bound with calcium is triggering the export of the calcium 2 plus ATPase. Eventually, what are these? These are proteins only, correct? They are proteins only. So these proteins will get exposed on the plasma membrane of the muscle cell. Now, these are the pump. So the ज्यादा calcium है जो आपके cytoplasm में वो आपको बाहर कर देंगे, ठीक है? They will be pumped out. Calcium, excess of calcium will be pumped out using the calcium 2 plus ATPase. And it will bring the normal concentration of the calcium 2 plus, which is the resting stage of your cell. If you remember in the previous slide, let me take you back. This slide. What is the normal calcium 2 plus का concentration यहाँ पे? 10 millimolar. So ये कैसे maintain होगा? With the help of your calcium 2 plus ATPase. Clear now?
Yes, ma'am.
हाँ. Okay. Now this is the structure. थोड़ा सा complicated. So this is the structure of the sarcoplasmic reticulum का membrane. Okay. On that top, you can see this calcium 2 plus ATPase is present. Okay. Now look at over here. You have got the, oh sorry. N terminal is there. Then how many transmembrane alpha helices are present? How many are present? 10. Okay, please remember. Okay, 10 are present. And what do we see that in this area? Okay, you have got the energy transaction, phosphorylation of aspartate and ATP binding. ये कैसे होगा? Because your alpha subunit is present over here. Okay, to which the ATP binding and the phosphorylation happens. And when this happens, there is a conformational change, which will result in the opening of the channel, where the calcium is able to bind and get transported from here to there. Okay, I'll show another figure, which will be more clear. Yes, this is the figure. Let's talk about this now. Okay. So this is your first stage. What is the first thing? The calcium is there. Okay. The calcium comes and binds to your calcium 2 plus ATPase. E is like enzyme का जो हमने दे रखा है, ठीक है? तो वो आपको enzyme state बताएगा. So what is happening at a time? Two calcium ions are coming and binding to its calcium binding site. Just now we saw the calcium binding site. See, here we have got the 4, 5, 6 and 8 together forms the calcium binding site. Okay. Now, calcium binding site is there. So what is the formula over here? Enzyme and 2 calcium binding sites are there. After this, the ATP will come and bind in the nucleotide binding region. N stands for nucleotide binding site or binding region, whatever it is. Okay, so we know that the ATP is the nucleotide. So it will come and bind over here. Okay, now when this ATP is present, what is the formula now? E1, calcium 2 plus twice and ATP. Correct? Now what had happened? ATP hydrolysis has happened. Okay, now what will happen? ATP जो है आपका, ATP, it will become your ADP plus PI. Correct? So this is your ADP and phosphorylation has happened over here to the aspartic residues. Did you get this point? After the hydrolysis, ADP phosphate is there. See, phosphate at the aspartate residues. Clear? Now, if you have seen from here till here, there is a conformational change. Okay. Are you able to follow, guys? Shruti?
Yes, ma'am. Yeah.
Okay. So ADP means hydrolysis ho gaya hai. Now, once the hydrolysis is happening, there's a conformational change. First thing is that ADP does not have affinity, okay, to be bound. No affinity to the enzyme now. So it gets released. And there is a conformational change over here. So what do we see? It comes and interact over here. Sorry. And the channel gets open. So the channels are open. So what will happen? The bound calcium is now released out. Okay. पहले क्या था हमारा इन कंडीशन था इन कैल्शियम इन हुआ है यहाँ पे अभी कैल्शियम आउट हुआ है नाउ दिस इज अ स्टेज E2 where you don't have anything. ना कैल्शियम है ना फॉस्फेट है ना ATP है. Okay, so now it will get closed and come back to its resting stage, where in which another round of binding of calcium will happen and the cycle will repeat. Okay, for a minute, just go through the entire 6 steps. Just see in which stage are you getting confused or any doubts are there. Yes. Any doubts?
Ma'am,
Huh.
ma'am, in the first figure, what does the A stand for?
Where?
The blue color structure.
Okay, this is again your domain, alpha domain, I think. This is alpha subunit when I was talking about, right? I'll show you here. See, you have got this energy binding, phosphorylation of aspartate and ATP binding site. So this particular A, B, C, not A, B, C, sorry. That A and what is the next residue they have said? P and then N they have written, right? So this is the area where your alpha subunit is present and these domains are present, globular domains. So where all this thing is bound. Clear?
Yes, ma'am.
Huh? This blue A you were talking about, right?
Yes, ma'am.
Okay. Anything else in the mechanism? Is the mechanism clear? So these, which is written on the top, if you see carefully, which is written on the top, ye bhi clear hai, right? So instead of protein, we just say that enzyme. Okay, enzyme bound with 2 calcium, enzyme bound with calcium and ATP. Here, from here to here, what had happened? Can you see the difference? The enzyme got phosphorylated, so it became E1P, calcium twice, and ADP. Then ADP was released out and calcium was out. What is left? Only E and P. So now the state is E2. Can you see the change over here? Now why they said E2? If you see the change in the structure of the enzyme, there is a huge difference is there, right? ये यहाँ से open होके यहाँ पे bind हुआ है. Now it has opened from the top. So there's a conformational change, and because of which now the enzyme is in second state. So ये E2 हो गया है. Then phosphate is removed by the hydrolysis. Only E2 is present and back to its even state. यानी कि क्या? Again it is back to open to this side. ये आपका inside वाला side हो गया. ठीक है? Clear?
Yes, ma'am.
Okay. And आपसे एग्जाम में कोई भी ये नहीं पूछेगा कि what is the mechanism this, okay? But what they can ask is कि पहले calcium bind होता है या पहले ATP bind होता है? पहले ADP रिलीज होता है या पहले कैल्शियम रिलीज होता है? So this is a thing which they can ask in the exam and whatever the multiple choice question they gave. So what is your answer? First is the, of course, the enzyme to which your calcium binds, and then ATP binds, ATP hydrolysis, ADP release, then calcium release, then phosphate release, then back to your normal enzyme. Can you do that? So what will be your assignment or your task to make a written notes of this? Like in 6 stages, you can just write it down. Of course, it is difficult to make the diagram, or you can listen to the video again and again, that is there. But agar prepare karna hai, for example, for the calcium 2 plus ATPase, okay, then we need the steps to be very clear. Abhi aapka kaisa hoga? Yes, I have understood how the calcium 2 plus ATPase is working. Okay, tumhe mechanism malum hai. But if the question is loud, after ATP hydrolysis, is it the calcium which releases, or it is the ADP which gets releases, or the phosphate is getting released? So what is the sequence? First is your ADP. After ATP hydrolysis, first is the ADP, then calcium, then phosphate. Okay? Are you able to get the point which I want to convey? Like what you need to remember in the mechanisms of these active transport?
Yes, ma'am.
Okay, good.
Yes, ma'am.
Now let's talk about the sodium potassium ATPase pump. This is the most frequently asked and the common pump to be to be studied and for the exam also. Okay, so it has got a Na plus K plus ATPase. It's a tetramer of alpha 2 beta 2 subunits, and इसी पे base numericals भी आते हैं, because it transports 3 Na plus out and exchange of 2 K plus in on the cost of the ATP molecule. So इसमें अभी formula क्या हो जाता है यहाँ पे? 3 Na plus 2 K plus 1 ATP. ठीक है? I'll give you here itself what was the question. I think once or twice only they have asked. So the question was, if 150 ATP was used, okay, how many Na plus and K plus were transported? So क्या आंसर होगा? सिंपल है, since you know this. So obviously, what will be the answer now? Simple maths.
450 Na plus
Correct.
and 300 K plus.
Correct. Simple. But for which you should know that it is 3 Na plus 2 K plus 1 ATP. ठीक है? Because option में कैसे आएगा confuse करने के लिए? 450 Na plus या 450 K plus. So the numbers are going to be these only. 450, 300, 150. But confuse करने के लिए ऐसे भी दे सकते हैं. Okay? तो आपको ऐसे भी options आ सकते हैं, and all the whatever the different possibilities. So based on that, like based on your memory or understanding, you have to select the option. Okay. Now, so it maintains the high concentration of Na plus outside and high K plus inside. तो जैसे कि अभी हमने देखा था, K plus आपका जो है अंदर ज्यादा रहेगा and Na plus जो है आपका बाहर ज्यादा रहेगा. Okay. This is the resting stage का concentration. हर एक टेक्स्ट बुक में थोड़ा थोड़ा अलग रहेगा. But what you can remember is that K plus is always high inside and Na plus is always high outside. बस इतना याद रखना है, ठीक है? Because people do get confused कि Na plus अंदर आ रहा है या K plus अंदर आ रहा है. So always that confusion is there. So what you have to remember is, if this is the membrane, ये आपका in है, ये आपका out है, then Na plus is always outside. K plus is आपका हमेशा inside at the cost of 1 ATP. ठीक है? यहाँ पे 3 Na plus 2 K plus at the cost of 1 ATP. This is what you need to remember. Okay, so inhibitors, this is also important. Obane and digoxin by inhibiting the ATPase activity. So they can just directly bind and block the pump. Okay. So now this ATPase also have two conformation. Again, if you have understood that, then this will be more easy to understand. So first one is enzyme 2 configuration, which has been phosphorylated and has high affinity for K plus, low affinity for Na plus. Dephosphorylated, it is your first enzyme state, has high affinity for Na plus and low affinity for K plus. This is something that you have to remember. Okay? बहुत confusing रहता है, बहुत मतलब बहुत ज़्यादा confusing रहता है. When you start doing the questions, PYQs, तब आपको realize होगा कि even though you know Na plus K plus ATPase, मगर जिस तरह से question पूछते हैं उसमें confusion आ जाता है. Okay, let's see the confirmation here. Now, so as I said, it's a tetramer, so alpha 2 and 2 beta subunits are present. Okay, now what do we see here is that always remember Na plus bahar jada hai, K plus andar jada hai. ठीक है, and we have to maintain that same. तो अभी हम क्या कर रहे हैं, 3 Na plus बाहर छोड़ेंगे और 2 K plus हम अंदर लेंगे at the cost of 1 ATP. I'm repeating again and again. So what is happening now? First is your 3 Na plus and ATP binding is happening. Okay. Now, then phosphorylation of aspartate is happening. तो phosphorylation कैसे होगा आपका? ATP hydrolysis से होगा. Correct? Correct? Now, so phosphorylation had happened. There is a conformational change. That time, your 2K plus is coming and binding. And Na plus is getting released. Okay. Now dephosphorylation is happening. Again, there is a conformational change. That time dephosphorylation हुआ तो of course phosphate आपका remove हुआ है. Phosphate के पीछे पीछे K plus आपका remove हुआ है. That's it. Clear? Should I repeat? Take your time and please do understand that this is really very important. Okay. The steps are important. कब क्या bind हुआ, कब क्या release हुआ, that is more important. Okay, who can explain me this? Shruti, would you like to try explaining the Na plus K plus ATPase mechanism?
Okay. So first in the pump, 3 sodiums go in along with 1 ATP.
Yes.
And all of them bind, and then aspartate is phosphorylated.
No,
So
before that, what should you say? Okay.
Sodium goes in.
Sorry, like I interrupted. You continue.
Yeah. So sodium and ATP bind, and then because of when the ATP binds, aspartate is phosphorylated. And ATP causes phosphorylation of aspartate, and then
after
ADP
hydrolysis. After hydrolysis, that is important. Okay.
After hydrolysis, yeah, yeah, and then ADP is released, and then that causes the potassium to come in.
No. See,
and then
when ADP, when ATP hydrolysis happened, it means what? ADP and this phosphorylation was there, right? The I didn't mention which residues. I guess it's aspartate only. Oh, time is getting over. So aspartate gets phosphorylation. Okay. Now this phosphorylation causes conformational change. Okay. Now.
Okay. So the conformational change causes the affinity for potassium to increase. So potassium is then and then the sodium is released, and then potassium binds, and then the phosphate is still there. So dephosphorylation happens, and that causes the conformational change. So when the phosphate residue which is left, that is released, and after that potassium is released. Correct. And then the enzyme returns back to its former state.
Yes.
Again.
Yes. So when this phosphate group is removed, the channel, this pump gets open for your phosphate. Okay. This is like पहले ऐसे open था, अभी ऐसे open है. I guess I will have another figure also. So I hope it's clear. Okay. Akshaya, Tanvi, what about you guys?
Yes, ma'am.
Okay, good. Okay, let's talk about like how it is inhibiting and all. So because of the transport of 3 Na plus outside and 2 K plus transport inside, a membrane potential is -52 -70 के बीच में हमेशा रहता है. ठीक है. Now, obane is potent and specific inhibitor of this Na plus K plus ATPases. It binds to the protein that is open to the extracellular side, locking in two Na plus ions, preventing its conformational change. यानी कि क्या, जब Na plus bind हो रहा होता है तभी ये आके भी bind हो जाएगा, and because of which it is blocked now. ठीक है. Now it is a cardiac glycoside and in lower doses can be used for medically to treat hypotension and arrhythmia. But of course, if you give in high concentration, it is going to cause a heart attack. Okay. This is the flower, okay, which I showed before. One second. This one, and this one, digitoxygen. They both are like, responsible for causing the heart attack. Okay. It is also known as the foxglove. It is used to treat congestive heart failure and strengthens the heart muscle contraction. But it inhibits the efflux of Na plus, raising the intracellular Na plus concentration. This activates Na plus calcium 2 plus antipoter in heart muscles, is increasing the influx of calcium 2 plus, which strengthens the contractions. ठीक है? So यहाँ पे these are the two which is affecting the Na plus concentration. Clear? Okay. So I'll be stopping here now, guys. Any doubts in this section which we have discussed just right now? So today we have discussed is the P class pump. Examples, we discussed calcium 2 plus ATPases, and similarly we have got this Na plus ATPases, Na plus calcium 2 plus ATPases. Okay, any other doubts are there in this section?
Ma'am, which are the flowers that the medicines are derived from?
This foxglove and what is that? I don't know the other one. Obane. Oh, one second. Foxglove. I don't think it is there in India. I don't know. I have not seen. और देखा भी होगा तो मैंने भी पहचाना नहीं होगा.
Okay.
I'm sure it will be there because see, in Ayurveda, this is what they have been trying, right, to treat the patients with the help of minute concentration of this. I can tell from my example, like when I was doing PhD, I was working on Dhatura. I don't know how many of you people know what is Dhatura. The scientific name is also Dhatura. Scientific name is Dhatura inoxia. Common language, it is Dhatura. This is that fruit which has got spikes. We put it to the Lord Shiva. If you remember.
Yeah, ma'am.
हाँ, कुछ ध्यान आया? हाँ, ऐसे कुछ रहता है, green color. After getting, so its flower has got white color flower रहता है, and ये color का flower रहता है, purple color flower is there. So I was working on this. ठीक है? So usually people use this as smoking weeds. ऐसे नशा करने के लिए रहता है, because it has got that effect. ऐसे जो opium या the drugs लेके जो effect आता है ना, those people who take hookah and all, अभी मुझे याद भी नहीं आ रहा क्या बोलना चाहिए, जो हुक्का लेते हैं ऐसे, just understand that this is also used as a drugs, like opium, heroin, hashish, ganja type का. ठीक है? So this is also that, but if you use it in a proper concentration, like minimum concentration, it acts as a medicine. ठीक है? तो जिसको अस्थमा का प्रॉब्लम है, who have got ऐसे कुछ लोग होते हैं जिनको सालों साल की खांसी रहती है, because of the infection, and see, infection is there, so obviously your blood count will have high concentration of eosinophils, because this will react when you have got infection. So all this Dhatura inoxia की जो seeds होते हैं उसको आप mildly गर्म करोगे, you take the inhale, okay, you take that smell, so this can treat this particular infection. But if you use it like very very high concentration, आपको पता भी नहीं चलेगा, because it doesn't have any taste. आप किसी के खाने में या कुछ भी करके मिला सकते हो, that person will die of heart failure then. Okay, so everything has got its side effects. Similarly, so I was working on this and I came to know that I still remember that if I use 140 microgram of the concentration, it can kill pancreatic cancer cell. Since it was my 5-6 years ka kaam, so I still remember that if I use 140 microgram of this protein, it can kill pancreatic cancer cell. Microgram is what? Nothing, right? But if you use in proper concentration, then it is going to have the medicinal value. Okay. Similarly, I believe that everyone is eating cabbage. ऐसा कोई है जो नहीं खाता है? Everyone eats cabbage, right? you might eat the cooked form also and the raw form also. Agree?
Yes, ma'am.
Obviously, right? सभी खाते हैं. अगर Chinese पसंद है तो आपने raw में भी खाया होगा. But if you eat the entire one cabbage, not 1 kg, one entire ऐसे बड़ा वाला cabbage, if you eat, okay, it is toxic to your body. It is toxic and it is well proven also. तो आप कभी भी देखोगे, if it is cooked properly, then well and good. कोई problem नहीं, because the protein it has, okay, the protein called lectin, okay, it has. अगर आपने कुक किया तो obviously हम उसको 100 डिग्री तक तो जलाए ही है. So obviously the protein loses its structure because it gets denatured, right? Now if you're eating raw, just imagine eating 1 or 2 cabbage entire, ऐसे कच्चा पूरा without taking salt also. Then it is going to be lethal to you. When I say lethal means that death can happen. ठीक है, तो हर एक चीज अपने अपने अच्छी क्वांटिटी में it is beneficial or it has got the medicinal value. Okay. Anything else do you want to ask? I hope you are not scared of eating cabbage now. Okay, then I'll be stopping here.